distinguished guests, ladies and gentlemen, welcome to an evening of research on the brain. I'm Kevin Milne of Very Little Brain, former fair-go man, now a carpet salesman. I'm thrilled to open this very special evening. Let me tell you why. A couple of years ago, I suddenly found myself with a tumour on my pituitary gland. It was a hell of a shock, and I'd never actually knew where the pituitary gland was up to that stage, but it was a nasty thing, and I was in the hands of some brilliant, brilliant men who got me out of trouble. The operation was astonishingly good. They rolled the tumour out in one piece, which apparently is a bit of a rarity. And they had fired off an email of the procedure to New York the next day. So I felt quite special about that. And when this tour began last year to go around all the main centres, we went to Auckland. In Auckland, we had, in the North Shore, had the biggest turnout on any brain issue in the history of the country. Over 1,000 people turning up. And right through the country. We went to Christchurch, Dunedin and Wellington. Fabulous, fabulous turnouts, as we see again tonight. And it was very moving. A week after I got back home, my wife had a brain hemorrhage. And I knew all these wonderful people who could help me through and guide me through a difficult time. And she's fine now, so that's why I'm here. I'm moved to be part of a group of people who I have a great deal of affection and admiration for. This week is the Neurological Foundation's annual Appeal Week, and this event is part of a national lecture series to raise awareness of the critical need for brain research funding. The Neurological Foundation has raised over 40 million to fund brain research in the last 40 years and needs you to help continue that work. We'd like to make special mention of the Foundation's 22-year collaboration with Mitre 10. They sponsor the Foundation's annual appeal. They pay for the production and print of the appeal envelope, and they help to spread the Foundation's message across New Zealand. Quite simply, without Mitre 10 and their passionate staff, the Foundation would not be in a position to have Appeal Week. Thank you, Mitre 10. They also are able to supply us with quite cheap saws and drills and stuff for the surgeons. I'm joking, of course. The Neurological Foundation is the only non-government neurological research funding body in New Zealand. Over 98% of our funding comes from donations from the public. The rest comes from trusts and grant bodies. Funding for brain research is needed more than ever to improve the outcomes for the growing number of New Zealanders being diagnosed with neurological disorders, me and the missus being among them. As our population ages, so too does the number of these disorders. And the only way to end neurological disease is with the best research. Fortunately, research is making remarkable progress, as you'll find out tonight from our panellists. Each of them has contributed hugely to the advancement of neurological research in their respective fields. Their passion and commitment to improving the odds for patients is very significant. The Neurological Foundation has funded much of their outstanding work, and it's critical that this research continues at a high level so we can understand and treat neurological disorders effectively. I encourage you all to join the Neurological Foundation and to help keep this incredible work going. Okay, let's get on with the evening as such, the one you've come along to see. I'm proud to welcome to the stage 3 of New Zealand's outstanding brain researchers. First, Professor Alan Barber. Please come up. Professor Winston Byblow. Associate Professor Bronwyn Connor. It was always my dream to end up on stage with such brains. A bloke that had to struggle twice to get school cert. Anyway, I'd like to start by asking a fairly wacky question, really. What do you know now that you didn't know 10 years ago? And I'll let you, perhaps Alan, you might want to start.
Oh, well look, when I only started training to be a neurologist, I'm a clinical neurologist, half my time I do clinical neurology, the other half I do research and teaching. I only started becoming a neurologist about 15 years ago, seems like yesterday. And when I decided to do neurology, I got a hard time from my cohort of people that had been through medical school with. And the reason why was that neurologists were all a little bit odd. We have obsessive compulsive tendencies. We were very good at examining patients with our tendon hammers and our tuning forks and everything else. And we could make the most spectacular diagnoses of these really rare and very unusual diseases, but there was nothing we could do. And I'm not joking. I'm a stroke neurologist, so I deal with people with stroke. And patients who came in with stroke, and I'm talking about 9,000 New Zealanders every year, were the last people who were seen on the ward round, usually by the most junior person, because there was nothing we could do. There were no treatments for stroke. And 10 to 15 years later, we now have stroke units, which didn't exist then. For every 16 people we treat in a stroke unit, we save 1 from death or disability. We now have clot-busting drugs. So for people who come in with a stroke where an artery's been blocked by a clot, we give drugs to dissolve the clot. And in the Auckland region, we're treating about 10% of our patients with clot-busting treatment. What we're doing something else, which is really cool in the last year or two, is for people where the clots don't dissolve, we put a catheter into the artery in the groin and feed it up to the brain and actually hook the clot out. And so that's still experimental, but we wouldn't have even dreamed of doing that 10 to 15 years ago. Am I too far away? The other things are that multiple sclerosis, a terrible disease. 10 years ago, there was pretty much nothing we could do. The beta interferons were just coming into use and they have a modest benefit. But in the last couple of years, we've got drugs that can basically turn off the activity of multiple sclerosis. So drugs like natalizumab, it can stop it in its tracks. Now, it's horrendously expensive. It's about $50,000 per year. And there are significant side effects. But now that we know that we can turn it off, it's only a matter of time before we find other drugs that are less expensive, hopefully, and have less side effects. We have drugs for epilepsy now that have fewer side effects that can keep seizures under better control. We're making major advances in conditions such as Huntington's disease with the development here at the University of Auckland with a genetic sheep model of Huntington's disease. And the more we know about things, the more chance we have of finding cures. So, I mean, there's no comparison that in the last 10 to 15 years, there's been an explosion in research that has made significant differences to people living with these devastating effects of neurologic disease.
Bronwyn, do you want to add to that?
Yeah, sure. So my field of research is stem cell research, and 10 years ago, one of the areas of research where we've seen huge advances is the discovery that actually the adult brain, or the adult human brain, has stem cells and has the ability to actually, the potential ability to actually repair brain injury and cells that have died through disease. Obviously the brain doesn't do that effectively, otherwise we wouldn't have a lot of these conditions that Alan's already talked about. Stroke, the brain would recover and we wouldn't have the huge number of people being affected by stroke. So what we have there is an organ that's trying to repair itself but doesn't quite do a really good job. And what's happened over the last 10 years is we really started to understand the whole process of how the brain can actually recover and regenerate, and the whole system that's actually involved, which involves the immune system and the inflammatory system and cues telling the stem cells where to go and what to turn into, and really just actually starting to get a really good understanding about just what the potential for repair is in the human brain. And we didn't know that 10 years ago. We believed that once you reach the stage of about 20, when your brain is sort of fully developed, you then sort of started to lose brain cells at quite an alarming rate. And it was sort of a slippery slope downhill to old age and dementia or whatever it was you were succumbing to. So the discovery now that actually the brain can repair itself, and actually every day our brains are doing a self-repair process. We have a population of stem cells, the richest population of stem cells in the brain is an area called the subventricular zone. And what they do, and what they're doing in everyone's brain right here at the moment, is they're travelling up a pathway called the rostral migratory stream, which we actually identified at Auckland University, and up into the olfactory bulb. And what they do is they make new olfactory bulb neurons. And the reason this happens is that because the nose is actually exposed to external stimuli, every 2 to 3 weeks, then the neurons or the brain cells in your nose actually die off. And so they are actually replacing those continually. So we actually have a self-repair process going on in our nose, which is useful, I suppose. But and also leads to a lot of really interesting observations which clinicians hadn't actually really been able to put together, which was in some disorders such as Huntington's disease and Parkinson's disease, patients will actually say that one of the first things they started to lose was their sense of smell, meaning that the self-repair process may actually have been deviated to repairing the brain cell loss as opposed to replacing the neurons in the nose.
And this research is leading the world, really, isn't it? I mean, this is all happening here. I'll get back later on, but I'm interested in why it is that New Zealanders are so good at this sort of research. But Winston, can you just give us your views on the last 10 years?
The last 10 years in my area, that sounds pretty incredible what they're doing, doesn't it? My area is perhaps a little bit more modest because I'm really interested in how the brain is involved in controlling movement. And you think about movement, and you think about, well, that's what your muscles are for, to help you to move. But in fact, if you think about how we're able to recreate the functions of the brain, and you think about the most powerful challenges that you can give a brain, for example, what is the brain capable of doing? Fundamentally, perhaps it can recognize patterns, it allows us to see things, it allows us to perform very skillful activities. And we have computers now that can mimic the way the brain is involved in, say, playing chess. And computers like Deep Blue that IBM invented can beat the world's best chess player. But think about how well machines are able to recreate movement. Remember all those science fiction examples of what the house of the future would look like with the robots moving around as if they were automatons? It hasn't happened, has it? We don't have a very good understanding of how we control our movements. It's an extremely complicated process, and it's intricately relying on commands and control from the brain all the time. Now we're better at understanding some of the important features, particularly how the brain responds to challenges when we're less able to move, and perhaps how movement can help, restoring movement can help the brain recover other functions, including cognitive functions and our intellect. But I think some of the gains in terms of knowing how the brain works in a way that would allow us to control such a complicated organism that's us in a way that moves purposefully and meaningfully through the environment. You're going to give me another 10 years on that one, right?
Well, that leads me nicely into the next question, because having looked back over 10 years, what are we going to know in 10 years' time that we don't know now? I know that's a ludicrous question, but I'm going to ask it anyway.
You want to start down at this end?
Yeah.
From my point of view, I still don't see robots walking around in the household in 10 years' time. But I'm really fascinated by the ways in which we're developing technologies to better visualize the brain in human beings. I'm lucky I get to work with whole human beings most of the time, and so in many respects, I'm very grateful when people participate in my research, because they're sort of donating their brain to me while they're still using it, which is very nice of them. And it's been impossible to understand how is that organ functioning, and how are the different areas of the brain relating to one another. And within the last 10 years, we've been able to visualize that very well, and the new technologies now are allowing us to see how one area of the brain is interacting with the other, perhaps compensating for damage. And I think that level of how is the whole brain working, and how are the connections changing, either in a useful way or in a not so useful way, I think we're making great strides in that area.
Bronwyn?
Yeah, I think we're going to start to see, hopefully in 10 years' time, the development of potential therapies that use or augment the self-repair process that the brain's trying to do. The other big thing that's coming up, and I think we probably will talk about this a bit later, is the whole field of reprogramming and what that's going to mean for the treatment of neurological diseases.
What's reprogramming?
So reprogramming is a really recent technique, and it was actually first developed by a Japanese group in 2007, where they showed that you can take a piece of human skin, overexpress 4 genes, and turn them into stem cells. So that is pretty incredible.
Yes.
It blows my mind. And so what you can then do from that, and the work that's led on from that, is actually really exciting. And I think the next 10 years we're going to see the idea of reprogramming really coming to the forefront for the treatment and understanding of neurological diseases.
Alan, of course, you're in the hospital. You have a different... Perhaps I should make this clear in case you didn't pick it up. We have here a neurologist with Alan who's working in the hospital with patients all the time, and our other 2 panellists are top researchers who spend most of their time in the laboratory. Well, when they're not touring around the world speaking at conferences.
It's very glamorous.
He says jealously. But yeah. Alan, next 10 years?
Well, I think, we cannot underestimate how important this discovery about the generation of new brain cells is. And now that we know that, it's amazing, isn't it? I mean, 5 years ago, 6 years ago, I was teaching medical students that, once you were an infant, that was it. That was as many brain cells as you were going to get, and it was all downhill from there. And that's not that long ago. It's only a few years ago. And so now that we know that you can generate new brain cells, it is only a matter of time before we reprogram skin cells to become brain cells. We can generate the sort of brain cells that are missing in particular diseases, and we can get them to go where they need to be. So I think that's the most exciting thing. I think the other really exciting research thing concerns something called brain plasticity, and the ability to encourage the brain, or surviving bits of the brain in people with stroke or traumatic brain injury, for example, to take over the function of the parts of the brain that have been lost. And I think that's something that Winston and I are working on in some of the research that we're doing. It's really exciting. The other thing that would be really nice to know is that, all these fancy things, I'd really like to know what causes Parkinson's disease. I'd really like to know what causes multiple sclerosis. We can generate brain cells from skin cells, but we don't know basic things like that. And unless we know what is causing diseases, we're up against it in terms of finding cures. So I would really like to, I would hope in 10 years' time we'll know what causes Parkinson's disease. There's a big study going on here in New Zealand and Australia looking at multiple sclerosis and whether vitamin D deficiency is causing multiple sclerosis in some way. You know that multiple sclerosis is more common the further away you get from the equator. So in New Zealand, it's more common in Dunedin than it is in Christchurch, than it is in Wellington, and it's least common in Northland. And one of the possibilities is that people in the deep south are getting exposed to less vitamin D. It'd be really cool if we can show that that's one of the causes for multiple sclerosis. So we've got all these amazing science things, but some really basic things like what causes Parkinson's disease, multiple sclerosis, epilepsy would be really good.
Is there much of a jump between finding out what causes these major diseases and finding a cure for them?
No, that's the really cool thing. I mean, take Parkinson's disease for an example. Knowledge of the brain chemistry, which we gained in the 1950s and 1960s, scientists at that time figured out that the reason why people got Parkinson's disease was because the dopamine secreting brain cells were degenerating. And so the dopamine, which is a neurotransmitter, it's the way brain cells communicate with each other. You get an electrical impulse that goes down the axon, sort of like this, gets to the end of the axon, spits out some dopamine. The dopamine joins onto the next brain cell. That's how they communicate. It doesn't take a brain surgeon to figure out that, you know, if the dopaminergic brain cells are degenerating, give dopamine. And so that's how drugs like Cinemet were developed and Matapar. And, you know, we've got fancy new Parkinson's drugs, but those two are the best. And so by knowledge of what causes something, by knowing what causes something, it's a major step towards figuring out how to cure it.
Before we go too far ahead, I want to go back to basics, really. Bronwyn, what is the brain? Can you explain what is the brain? What does it do? What's its function? What does it do?
So
Not too long about that,
but
but
easy question.
fairly briefly.
Okay. How long is a piece of
Brain
string?
101.
Have you brought one along or something?
No.
Can we see yours?
No. So, I mean, the brain, if I was to summarise in my thoughts about what the brain was, it's the central processor. Okay, it controls everything. Obviously, I'm biased, but I think it's probably the most unique and exciting organ that we have in the body. And it can, I mean, if you think about it, if you think just very basically, it's made up of neurons that communicate to each other, as Alan just beautifully showed by wiggling my mic in front of me, by electrical currents spitting out a chemical neurotransmitter and talking to each other. And then around them are supporting cells called glial cells that actually provide food for the brain cells and support and structure. And you have lots of different regions that obviously have very selective functions. And those functions are just so varied. So they range from the real basic things like how keeping your heart beating and breathing and movement control, as Winston said, through to hearing and speech, reading, and then through to really quite complex things such as personality and thought and dreaming and all those sorts of things. So you're talking about an organ here that just really does absolutely everything that is required for us to exist and also for us as an individual.
Winston, what happens when the brain gets diseased or injured? What's going on? What's going on? You know, we're talking about those big causes like dementia and stroke. What happens there?
Different things with different conditions, obviously. But to follow on, to answer the question, but to follow on with how Bronwyn was describing the brain, one of the really interesting things about the brain and the whole central nervous system, so the brain including the spinal cord, is it really doesn't like change. Now, if you think about that for a moment, what that means is that when things start going wrong, like in a neurodegenerative condition like Parkinson's, because the dopamine production area is slowly dying away, the brain knows to compensate, and it will compensate so that things stay the same. And all of that is always going on in the background. The brain is always compensating to keep things on an even keel. We call that homeostasis, equilibrium, stay in balance. So when there's an insult or neurodegeneration or something like this, you get this immediate reaction from the brain, sometimes at the cellular level, sometimes across thousands and thousands of millions of cells, saying, how can we compensate for this? So it makes it very difficult sometimes when you're thinking about treatment, like Alan has to do, because the symptoms that might now be in front of you as a result of this condition, some of them could be related to the original problem, and some of them could be related to the compensation process. And so sometimes what you can inadvertently do is go trying to address a neurological condition by alleviating a symptom, which is actually a compensation and a way of keeping function as high as possible. But it's interesting to step back and think of the brain at that sort of a big level of this thing really doesn't like change, and it's always going to try and compensate to keep things balanced. Regardless of the particular type of insult or neurodegenerative condition.
Yeah.
At a big level.
It is astonishing, isn't it? 1 in 5 New Zealanders will suffer a brain disorder at some point or another in their lives. And another fact that I heard, which I frankly couldn't believe, was that more people have brain disorders in New Zealand than combined have cancer or heart issues. It's astounding. Why, Alan, why do so many people in this country have brain disorder issues?
Well, I guess the first thing is, I mean, the brain is just the most amazingly complex organ. It only weighs 1.5 kilos, give or take. But it does, you know, as Bronwyn said, it controls all other organs. It's sort of, it is the central processor. And that complexity makes it vulnerable to disease. The other thing is that, one of the other things is that, you know, for something that has a consistency slightly firmer than jelly, it sits at the top of our body. And while it's encased in the brain box, the skull, it's susceptible to injury from traumatic brain injury. And large numbers of New Zealanders are affected by traumatic brain injury every year. Another thing is that unlike other organs where the organ stores energy in case there's an interruption to the blood flow, the brain doesn't. The brain is acutely sensitive to changes in blood flow because it only gets its energy in the form of glucose from the blood. And so if you reduce your blood supply or stop your blood supply to the brain, within a few seconds you'll lose consciousness. Within a minute or so you'll lose consciousness. If you've had a stroke where an artery, which is like a hose that takes blood from the heart to the brain, blocks, the brain downstream from it will stop working within a minute or two. And if you don't restore that blood flow, that brain will die. And that's what affects 9,000 New Zealanders every year with stroke. The other thing is that, as Bronwyn said, while we now know that you can generate new brain cells, they're not enough. And so, you know, you can live without a kidney. You can donate your kidney to someone else. You can take out half or 3/4 of the liver, and the rest that's remaining will just get bigger. You can transplant your heart. You can transplant your lungs. You can transplant your pancreas and your kidneys. You can't do that with your brain. Don't listen to people who say you only use 10% of your brain. You need all of your brain. And so it's not like the other organs where you can do without a bit. You need it all. So these properties of the brain make it particularly susceptible to degenerative changes, to vascular insults, to trauma.
Does the level of brain disorder in New Zealand relate poorly to other countries? Are we particularly prone over here for some reason or another? Or do we just represent a large amount of brain disorder right throughout the world?
Certainly throughout the Western world, we have similar sort of rates of brain diseases elsewhere. Our stroke rates are a little bit higher because we have more risk factors in our population, but pretty much the same. Is
there anything we can do about cutting back the amount of disorder? For example, if we look at areas of sport, and I know there's a lot of issue about the fact that we expose ourselves in rugby and what have you to injury, I don't know, diet or Is there anything that we can do to help?
How do you keep your brain healthy? It's a no-brainer, really. It's, the first thing, use it or lose it, okay? Coming to things like this and learning new things and, exercising and stretching your brain, that's good for it. Socialization, being with other people is also really important. People who are isolated are at greater risk of problems like dementia. So use it or lose it. Feed it properly. You know what you feel like, I know what I feel like, at 6 o'clock in the evening or 6:45 in the evening when you haven't eaten since, lunchtime, you feel a bit sort of jaded and tired. You need a good, healthy diet, a good balanced diet to keep your brain healthy.
Alcohol a problem?
A little bit of alcohol's okay, but, the other myth.
Alan might be hungry.
Yeah. No, I'll only talk more if you give me that. The other...
Alan can wait.
The other big myth, you hear about, sort of wine's good for your heart and it's good for prevents strokes and things. And that's true. A glass or two of wine is good, but what the press don't tell you is, 3 or more glasses of wine is bad for you. It increases your risk of stroke and heart disease and can cause cognitive problems, memory problems. So, a little bit of everything is good for you.
Well, I'm going to stop you now, Alan. You're attacking the press. Tonight we're talking about neurological research. Winston, how did you begin that research journey? Where did it all start?
Oh, Kevin, a long time ago. Remember what Alan just said about wine and memory? With any type of research, whether it's brain research or not, it's because you're curious. But like I mentioned, I'm interested in movement. I'm interested in how we control movement. And so I became very interested in conditions that limit the ability of somebody to be able to move, particularly as they get older. Because, like I said, it's this unique capability that we possess to be able to control this wonderful body that we all have, not mine. So we've got this capability, but we see that it's acutely affected with different neurological conditions, and it's a complete mystery as to how to go about solving that, depending on whether or not we're talking about having a weak side of the body after a stroke affects the other side of the brain, because this side of the brain controls this side of the body and so on, or if we start to experience a constellation of symptoms because a part of the brain is dying off, like the substantia nigra in Parkinson's disease, which sort of slows movement down. So when you go about as a researcher thinking about how am I going to help contributing to solving a problem like that, you sort of need to look in 2 directions, I think, and this is my personal view because you asked me. I look towards Alan in neurology, and I try to understand what it is that he can offer his patients and what is it that's lacking. And I have to look, I think, so that's looking ahead and then looking backwards to people like Bronwyn, who are making the discoveries in basic science that are going to lead to possible new treatments. And I try to work in that gap where, I'm looking in both directions. It's like your mother tells you to do before you cross the street. That's what I'm doing, to try and take some of the more exciting advances from the bench and seeing, will these translate in a way that people like Alan can use them? So that's how I see where I'm positioned and what I try to do.
Yeah, Bronwyn and Winston, did you both want to go through a stage of wanting to become neurologists or work in hospitals and work with patients at some point? Or what tipped you over to say, well, I'd rather actually be working in a laboratory finding stuff out rather than helping people get better?
Yeah, I think I particularly had a feeling quite early on when I was going through university and starting to look at either I was going to go to med school or I was going to go and do a PhD. And obviously my interest was in looking at brain disease and how to treat brain disease, and I think, you know, as Alan mentioned before, the frustration that he must feel all the time, that a lot of the patients he sees, he can't do anything for them. And I didn't think that I could actually do that job. I think it's an incredible job that he does. I wanted to actually be able to go and try and find things to make people's lives better. You know, and eventually maybe something in several years or a decade down the line, something that I have had a hand in will actually go to clinic, and that to me would be hugely rewarding.
Do you ever get the sense that all the work you're doing is not being taken on board by those who are working in the hospital?
You're not listening to me.
Oh, I'm going to give Ellen a chance to have a go back there.
But yeah, I mean, we, as Winston just said, I mean, I'm sort of on one side, Winston's in the middle, and then there's Ellen. I'm quite basic research. Basic research with definitely a look at the holistic point of view. So every research, all the research that we do in my group, always the thought is, would this have a clinical potential? There are a lot of good questions that need to be asked just for the sake of asking questions, and sometimes we ask those, but a lot of the times I want to see a therapeutic potential. Maybe not immediately from that study, but maybe, you know, several iterations down the line, but it will be something that eventually could go into clinic. But I'm sort of the initiator, and there's the middleman to transition it through, and here's the end.
And Ellen, you're at the end there, so do you ever feel that there's not enough work going on at the coalface to bring you new information and good new research, or are you impressed as I am? I suppose you've got to say this now.
No, I am.
Is there much more room for more research?
Oh,
of course.
Yeah, what do we know? We don't even know what causes Parkinson's disease. Of course, there's room. You know what the really cool thing is, though, is that we actually work together. That seems like a really obvious sort of thing. So, particularly Winston and I, we do research together. You know, Winston comes in and studies my patients, and that's actually quite unusual. And we do it here in Auckland better than almost anywhere else in the world, I think. And that's, I think, one of the reasons why we're so good at brain research. That New Zealand's too small to have silos where basic scientists work just doing this, and movement neuroscientists work over here just doing this, and clinical researchers like me do clinical research. We're too small to do that. We haven't got enough money. We need to do our research smarter, and so I think we're actually doing that. That's one of the really cool things about, you know, being in brain research here in New Zealand, is that we all work together.
Yeah. What have been the biggest changes in brain research over the last decades, do you think?
Oh, has to be the stem cell stuff. That's just amazing. The generation of new brain cells. You know, I cannot emphasize how important that is. I think also
Can you explain to those of us who are not necessarily scientifically minded just what's happening in that regard? Why are you so excited by stem cell research?
Well, I think Bronwyn, she's the one that's doing the research.
Bronwyn.
I'm just amazed by it.
No, okay. Bronwyn.
Well, as I said earlier, I mean, it's just the discovery that we can actually, our brain can actually generate new stem cells. So if we take it to the clinic, where, you know, Ellen has patients with stroke, it's a nice example, and, you know, we're very limited with the therapies that we can provide a patient with stroke, and we do see, and Alan could probably talk a little bit more about this, and so can Winston with the work that they do, that rehabilitation improves the outcome of the patients, and certain techniques that they use can improve the outcome. And that's about plasticity, the brain plasticity. But also what might be happening there, and we're starting to get the idea, is a self-repair process, okay? But as I said before, it's not enough. So in two areas in our brain, and we have stem cells, and they're left over, and in one region in particular, the subventricular zone that I mentioned before, is left over from development, when as an embryo you start to form the neural tube and it folds it in itself and eventually makes the brain. And so those are leftover, call them leftover embryonic stem cells that are now in an adult brain. So we now call them adult stem cells, okay? And what's really cool about them is that, so most people probably in the audience have heard about embryonic stem cell research and the debates and the moral issues. And a lot of people are probably waiting to see when embryonic stem cell research is going to go into clinic. And the thing that's holding embryonic stem cell research back from going into clinic is actually what makes embryonic stem cells really, really cool. And that is this term called pluripotency. And what it simply means is that an embryonic stem cell, because it is the building block cell, so when you have a fertilized egg, that fertilized egg is full of embryonic stem cells, and they can turn into any cell type and any tissue in the body, which is really incredible. So for researchers and scientists, embryonic stem cells, you can take an embryonic stem cell and you can turn it into a liver cell or a cardiac cell or a brain cell. And so that's pretty cool, okay. Yeah. But that's what they're there for. But because they have that potential, they also have the potential that when you want to use them therapeutically, we don't go and use embryonic stem cells therapeutically. We turn them into the cell type we want them to be. So if we're going to use them for the treatment of brain disease, we will take the embryonic stem cell and we will tell it through chemical signals in a dish that we want it to become a brain cell. What we need is 100% of those embryonic stem cells to go, right, I'm going to become a brain cell. If one of them decides to revolt, I suppose, to be the odd one out and decides, no, I'm just going to stay being an embryonic stem cell. I didn't get the message. That stem cell has the potential to turn into a tumour. That's what holds embryonic stem cells back from therapy at the moment. So they haven't worked out how to get 100% production. So the adult stem cells that we have in our brain are really exciting because they're not pluripotent. They can only turn into brain cells. Okay, so they're sitting there, they're still a stem cell, but all they know how to do is to turn into one of the 3 different types of brain cells that we have in our brain. So that's really cool. So they can then go out, and there's studies that we've done and other groups have done that show that when you have an injury to the brain, they divide, they make a lot more of themselves, and they proliferate, and then they actually migrate and they actually direct themselves into the area, and they start to make new brain cells. That's where it doesn't actually, but then it doesn't actually go according to plan. So the immune system, which gets kicked in because you've got cells dying, the immune system kicks in, comes to mop up the dying cells. It's sort of like a bit of a, it's a clean-up crew. The immune cells are actually what tell the stem cells to come in. The stem cells also respond, oh, yep, cool, we've got an injury here, we need to come in, yep, we're going to make new brain cells. Unfortunately, when you have a stroke or if you have traumatic brain injury, for example, or another neurological disorder, that immune response turns into chronic inflammation, and that kills those new brain cells off. So this is where we're at in the field. It can do it, but we have to work out how to actually stop the brain or the immune system from killing off the new brain cells.
And which of those major areas of disease, Huntington's, Alzheimer's, Parkinson's, which is stem cell research likely to help the most? Do you know? Or is that pretty even across them?
No, there are certain diseases that will be more effective. Unfortunately, the closer you are to the source of the stem cells, the more likely it is that the self-repair process will happen. So what I'm saying is that the stem cells, they respond to their environment. They respond to cues and signals, okay? Otherwise, they're just sitting there going up to the nose, doing their normal thing. So if you have an injury to the brain that's too far away from that source of stem cells, they'll never get the signal. Then they'll never know what to do. So you're going to have a brain injury or brain disease, make sure it's close to the stem cells as well. So that's an unfortunate thing at the moment. But what we're trying to do is, but you can also, obviously with the embryonic stem cells, if we can get over those issues and the reprogramming stuff, reprogramming cells, you can actually look at transplantation as well, transplanting new cells. Yeah.
Winston, you work and would need to work, of course, with real people, real brains in your research.
Yeah.
Is it hard in New Zealand to get people to play a role and assist with research?
Thankfully, no. As I mentioned, they're very generous. They're generous with their time. They're generous because, as I said, they're donating their brain usually while they're still using it. that is extremely valuable in terms of getting information about how some of these self-repair and plasticity processes are going on, and particularly stroke and Parkinson's disease are two of the areas. How is that possible? How do people find out? How do people engage with us? Because it's difficult. It's difficult to do research with whole human beings. You often feel like as soon as you think you understand what's happening with one person, you get your next person in, and they're so different. Everybody's different because maybe no two strokes are exactly alike. They've affected a slightly different part of the brain because of where the blood supply was interrupted, for example. But the university and the Center for Brain Research, and with the help of the foundation, we've started a volunteer database. Anybody can go online, register their interest saying, I'm willing to be a participant. And without that, it would be so much more difficult to do the kind of research that we do. So I get people to come into the lab or come into where Alan works in Auckland City Hospital, and they can pretty much engage with really what I think in some of these areas are pretty much the cutting edge research that we can do with whole human beings non-invasively. We're not
Alan, you
not cutting into anybody.
Yeah, I mean, I'm a clinical researcher, so I research people. And it's humbling how people are so willing to participate in research. You know, over the years, there have been controversies about medical research and things, but Kiwis are really, really generous. And, you know, if you come in with a stroke, if you've got Parkinson's disease or one of these other terrible things, people often feel, well, as, you know, at least I can help somebody else. And so people are very generous. We almost, hardly anyone ever declines to take part. We always get their consent, obviously. And it's just humbling. It's
The classic example of this has to be the brain bank, doesn't it?
Well, that's exactly. I mean, that's
where people leave their brains after they die for research.
Yeah, that's
who's best to tell us about that?
Well, that's amazing. I mean, so the Neurological Foundation, with your support, supports the Neurological Foundation of New Zealand Human Brain Bank, which is run by Richard Fall across at the medical school. And it sounds pretty ghoulish, but this is just one of the most amazing research resources there is in brain research in the world. It's probably the best brain bank in the world. And so this is people who are afflicted by diseases like Huntington's disease and Parkinson's disease. And we study people clinically in the clinic with their disease. And so we get the brain scans and the blood tests and the clinical examinations, and all the information's recorded carefully. But unfortunately, some of these diseases are fatal, and other people die from other causes as they get older, and have left instructions for their brains to be collected. And it sounds ghoulish, but it's the most amazing resource, so that researchers at the medical school can actually look at the brains, go back to the information we've had from the scans and the clinical information that we clinicians write down, and try and put it all together.
Mm-hmm. And in fact, people overseas, researchers overseas, are now using our brain bank as a facility to get information.
Yes, I mean, it's a really high quality, very important resource.
So you'd urge us all to consider the possibility of
donating. I'd urge you all to, because, you know, I guess many of you are affected by, in some way, either yourselves or your families with brain disease. And, you know, if you ever get approached to take part in clinical research, you know, think about it. As a clinician, I know that people who take part in research trials tend to do better than people who don't take part in research trials. You don't get seen by the professor of neurology on a regular basis if you've got a routine something. And so it's a good thing to do. Yeah. In fact, it's hard to get through the stroke unit at the hospital without actually getting put into a study. You know, we have arguments between Kathy Stanier over here, one of our scientists, and some of the other researchers, because we can only put a person in one research study at a time.
You're mentioning Kathy. I listened to a wonderful presentation from Kathy Stanier yesterday in Parapromu about plasticity, and that word's come up already a few times tonight. I think we need to explain what it is. Something to do with flexibility, isn't it? Can one of you take that on?
Yeah, it's most of, well, everything that we remember and everything that we've learned is occurred because of forming connections between these neurons in the brain. And the information is contained in those connections. And plasticity allows us to form new connections. Sometimes it also allows us to change the structure of the parts of the neurons that are capable of forming those connections. It can help us turn down, unweight connections that we don't want and promote new connections. So it's all in the bonds as opposed to in the cells themselves. And it's really unique. The fact that our brains remain plastic, not only at the time of development, but throughout our whole lives. We can demonstrate plasticity in the adult brain quite easily and convincingly now. We haven't quite figured out how to completely harness it and get as much of it in the areas that we need it when we want it, but that's what we're trying to solve.
It's a really difficult concept. I mean, each neuron, a brain cell, connects with thousands of other brain cells by these axons and dendrites and things. And, you know, plasticity is going on in all of our, well, hopefully it's going on in all of our brains at the moment, because that's how we lay down memories. So certain axons get stimulated and they make new connections, and the more you rehearse something, the stronger those connections are. And when you forget things, the reason why you forget things is the connections are unmade. And so that's what happens with plasticity. And from a therapeutic point of view, what's really cool, well, hopefully in the next 10 years will be really cool, is the ability to get the brain to make connections to compensate for the bits of the brain that have been lost. And so that's what we talk about, harnessing neuroplasticity, to get around the damaged area by getting other brain cells to take over the function.
Bronwyn's talked about her terrific work in stem cell research. In fact, do you want to just talk a little about your publication, well, Stem Cell and Regenerative Medicine publication, which is extraordinarily well received.
Yeah. So I talked a bit before about reprogramming, and this is the work that came out of Japan in 2007, where they took skin cells and put the genes in and turned them into embryonic stem cells. And so that's opened up a whole field of research known as reprogramming, and that's across not just neurological diseases, but across a lot of different situations as well. But so in my group, we were sort of watching this field and thinking looked pretty exciting stuff. And we decided, though, that for the issues that I told you about with embryonic stem cells, so the pluripotency issues, we didn't actually really want to take the skin cells and turn them back into embryonic stem cells, because you're going to face the same issue of pluripotency and the same potential tumour formation. So we decided that what we actually wanted to do was to take adult human skin cells and turn them into an immature brain cell, or what we call a neural precursor cell, which is a stem cell that is starting to become, it's not a really basic stem cell. It's sort of starting to become a brain cell. Okay, so it's, and we thought, okay, well, we'll give this a go. And so we decided, with some collaboration with a developmental neurobiologist at the University of Melbourne, we sort of sat down and scratched our heads and thought, what genes should we put in there? And we had a look at what actually happens during development, and the first two genes that come on as the neural tube or the brain starts to develop in an embryo are two genes called SOX2 and PAX6. And so we put those two genes in, and lo and behold, we could take adult human skin cells and we can turn them into immature brain cells, from which we can then generate all different types of neurons and glial cells from the body. And we're the only group in the world to have done this in adult human skin.
You talked about linking up with the Australians and that. Do we do that a lot? Are we?
Yeah.
Is the scientific world, is it all pretty close now? Are you on the phone to?
particularly with Australia. I mean, it's a small world. It really is. I've just come back from 4 weeks overseas in America and Germany, and we all work together. But the really exciting thing is, as Alan was saying about, what causes Parkinson's disease? Well, that is a really key question. So what we can do now with this technology that we've developed is that we can take skin cells from patients with Parkinson's disease and make these immature brain cells and then grow them up into mature dopamine cells. And we've shown this. We can do this in the lab, and we can make a huge number of dopamine cells, and we can start to watch what's different in the dopamine cell that we've made from the Parkinson's patient compared to a normal dopamine cell. And so we hopefully might be able to answer some of Alan's questions.
Well, and Winston, with Parkinson's, of course, there's this great line, isn't there, that once you've met one person with Parkinson's, you've met one person with Parkinson's. And how do researchers deal in a situation where there's such enormous variance as each of these patients report in?
Yeah, it's remarkably difficult, and Parkinson's is a great example. And just last week in Sydney, there was the Movement Disorders Conference, where there's big representation from neurologists all over the world, and a lot of them from New Zealand were there, and neuroscientists. And it was put very nicely by the best neurologists in the world who've been thinking about Parkinson's for their entire career, some of them 50 years, and they're basically saying, oh, we're right back to where we started from. And we talk about 50 years ago having that, discovering the importance of dopamine, that the dopamine cells are dying off, and what they call the L-DOPA moment, where they discovered that you can give levodopa to patients, and that will help replace the loss of dopamine. But here's the problem. The dopamine replacement therapies, and they've evolved over the years, and Alan's mentioned some of the most popular drugs. Not everybody responds in a uniform way. And the symptoms of Parkinson's disease usually begin as motor symptoms, like I'm the motor movement chauvinist. I see the movement things first, and you see the movement symptoms first. And so you try to treat the movement, the motor system. But what ends up happening is that too much dopamine goes to the areas that are actually not only involved in movement, and sometimes you can end up producing too much movement, and that becomes another problem, but it can also start affecting the cognitive system, because now it's being flooded with dopamine. It didn't have a dopamine problem, only the motor system did, or the emotional system. And different individuals are responding in a different way to this input, artificial input of dopamine. And so it was really illuminating to see how neurology was really putting the call out in a theater just like this one to the scientists, saying, help us solve this problem. And so even people like me who work with now people with Parkinson's disease, and my PhD student Haley right there, she's very interested. She's supported by the Neurological Foundation, by the way, with her scholarship. And she's come up with a really good idea of which genes might be most important to understand a few candidate genes that are really important for how dopamine is used and taken up in the brain, that might give somebody a potential fingerprint of whether or not they're going to respond favorably or unfavorably. And that kind of information in the hands of your Allens of the world can then be used to help say, for you, this might be the right drug, but for you, it's not going to be. We're going to try this instead. That's how you treat that problem. It's not going to be with herd medicine, one size fits all. It's going to be, how can we break this down in a way that gives us what we call biomarkers that are suitable for the individual person to know how they're going to respond. And we're trying to do the same, take those same principles after stroke and say, not one thing is going to help everybody with stroke. What can we do? What can we look at in the person's brain, the individual's brain, that's going to tell us how they're best going to respond and to what type of intervention?
Alan, 5 years ago, I think you were appointed into this, the chair that you're in at the moment, the Neurological Foundation Chair of Clinical Neurology. What are your highlights so far?
Phew, it's gone.
Just give us your top one.
Two.
Two.
Maybe three. Two. First, when I started in the University of Auckland, which is a major university in the world, there was no academic neurology. The university paid me for 4 half-days a week to look after the teaching, but that was pretty much it. There was no academic neurology. And so the development of the chair has allowed leveraging from all sorts of sources, and I don't know how we've done it, to have a full professor. We've got Kathy Sanera, fantastic movement neuroscientist. We've got Jennifer Pereira, who's a multiple sclerosis neurologist, both senior lecturers. We have the Douglas Neurological Foundation Fellow as a junior neurologist doing research. For a while, for 3 years, the Stroke Foundation chipped in with a Stroke Foundation Fellow, which was a scientist. We have a steady stream of international and national students, PhD students, medical students, coming through the lab and the unit to see what we're doing, to learn the techniques that Kathy's doing. And so we've gone basically from nothing to quite a significant unit in a relatively short space of time. And I have no idea how that happened. It's sort of, that wasn't what it was supposed to do. So.
Is Auckland now the centre of brain research in New Zealand?
Yes.
Bronwyn, you probably disagree with that.
No, yeah, it is.
Yeah,
I mean, there's really good brain research done at the University of Otago and Dunedin, and there are pockets of brain research, Parkinson's research in the Vanderveer Institute in Christchurch. So there are, but you know, we have at the centre of brain research. That's the other thing, is that the other major thing that I wanted to do when I took over as chair, or became the chair, was the gulf between the medical school and the hospital. It was only Park Road, but it was actually very wide. I think one of the things that we've been able to achieve, myself and Kathy and others, has been to try and break down that divide. And so now we clinicians, the neurologists and the neurosurgeons, and hopefully increasingly the neurosurgeons, are working more with our scientists. As I said, we're working together now. And that means that our scientists, we're making sure that our scientists are asking the questions that you want answered and having a more therapeutic focus. So, I mean, that's been the other major achievement.
What are the biggest obstacles to good research in this country?
Funding. I know it sounds obvious, but it really is. There's a lot of good research going on. All of it's important. We need answers at every level. And we simply can't do it without money. It's not like the money's being used to train the researchers of tomorrow. They're doing their studies. It's paying for scholarships. It's paying for working expenses. You know, it's not paying for a lot of business class travel or anything like that. That's certainly not the case. It's simply getting the work done and getting it done, I think, quite efficiently and cleverly, like Alan alluded to earlier. And one of the things about being in New Zealand is sometimes you think, do you feel like you're at a disadvantage being so far away or in a small country, maybe with less resources and less money? Possibly, but sometimes it allows you to work a little smarter. You have to be more resourceful. So I think what you can be sure of when you're donating to the foundation, for example, is that the money's being used. Not only is it being used well, it's being used very efficiently, and it's all going to solving an important piece of the puzzle.
And I mean, the foundation, I mean, it pays for me now, but it's paid for me from the beginning. You know, I was a Chapman Fellow, the Neurological Foundation Chapman Fellow. So when I had just finished my neurology training, the foundation paid for me to go and do a PhD at one of the top stroke centres in the world. And the foundation has supported me subsequently with research. And you were...
Yeah, I was the same. I was the first postdoctoral fellowship winner. And so I did my postdoctoral fellowship over in the States in gene therapy and in gene therapy for Parkinson's disease, which is now actually going into clinical trial, which is pretty exciting. And have since funded me. And I would say probably 80% of my PhD students are funded through the Neurological Foundation.
So the foundation is very clear and they're not paying me to say this, but is very clear in identifying brain researchers when they're junior and, supporting them as they develop their careers. And I think that's one of the foundation pays for research as well, but that's one of the major things the foundation has done. And that's another reason why neurological research is so powerful here.
Well, there are plenty of other questions to be asked, I think, but I think that's not a bad note to end on. Thank you very much. And I think we've probably got time to put up some questions for the audience. Can I just ask, though, that no more than one question from one person, and please don't see this as a free consultation. Make sure that your question is a broad one that would be of interest to most of the audience rather than just yourself. Okay, so we're going up the side there. The first question is...
Good evening. My question is very simple. Is there anything known as mind and how it is related to brain, or does it reside inside the human body?
So the question was, is there such a thing as the mind, and where does it reside? Is that correct? The mind, over the years, has exercised philosophers. Where is the seat of the mind? But the mind is clearly in the brain. And in particular, it's a very abstract concept, but it is something that resides within the brain, along with our personality and our emotion. That's where it gets generated from.
In how far have tools such as cerebral imaging become useful in neurological research? And what is the application, apart from treatment of motor neuron problems, in cognitive or behavioral syndromes such as anorexia nervosa?
And so I'd go down to the X-ray specialist, the radiologist, with 2 forms to get a scan on a stroke patient, because he'd always tear up the first one. And you'd pull out the other one. No, I really want one. And we were sending patients to Australia for MRI brain scans. This was 15 years ago. And now we have 4 MRI scanners within 100 metres of Auckland Hospital, give or take. And there are multiple scanners within Auckland. And it's allowed us to actually look at what's going on in the brain. Unlike other organs, you can't open up and have a look particularly easy. Very occasionally we do brain biopsies, but brain imaging has revolutionised how we approach people with brain disease, particularly with things like stroke and brain tumours and traumatic brain injury. We're getting to the stage now where there are new fancy brain techniques that probably Winston better talking about because he uses them in research, where we can actually look at the function of the brain. So you can put someone in a scanner, get them to squeeze a ball, and you can see the hand area of the brain lighting up, and you can do that with different activities. And so we're now not only able to see structure, if someone's had a stroke or is there a tumour, we're actually able to start seeing function. And it's another really exciting development.
Yeah, and that certainly does apply to cognitive functions as well as motor functions.
Yes.
So you can ask people to imagine things and see which areas of the brain are involved in imagining the future or the past, or thinking about doing activities, or just remembering events. So we can start to visualize the function of the brain for some of these more abstract, higher order activities that could help treat conditions like you mentioned, anorexia nervosa, and other conditions that are really more higher order problems that have been traditionally very difficult to understand what's happening in the brain.
Obsessive compulsive disorder.
Exactly. Yeah.
I can imagine there's a lot of pressure to get stem cells out of the clinic, but I remember in the 90s I was looking at a book somewhere, and it's an image that has always stuck with me since. Is somewhere overseas they did a stem cell experiment on someone who had, I can't remember what they had, but this person died down the track, and they did an autopsy and found they had fingernails growing in their brain. So yeah, I mean, to me it showed just how important it is that when you're saying to get them 100% 100% brain cells that they are. Just a question for Professor Byblow. At the beginning, you were talking about how you see people have brain diseases and they also come in with some other sort of signs which you have to be really careful to tell that they're not, you know, that you don't, you said you didn't want to treat those signs, which I guess you would normally treat in someone, but you didn't want to because it was a brain reprogramming. I just, if you could.
Yeah, there's been a lot of information in the last hour, hasn't there? So you were picking up on that idea quite rightly, that the behaviors or the symptoms that somebody might have at the time that you're studying them, they could be related to the original problem, like brain injury or neurodegeneration, or they could now be reflecting the compensation to that. And you can make the mistake of actually trying to address something that's actually compensating in a functional way. Now, obviously, we think very carefully about what are the compensations and what are the original causes, and you're trying to understand what's the original problem as the researcher. But it's just interesting to remind yourself that that process of compensation occurs spontaneously and automatically, and it can come from areas of the brain that seem very remote to where the original insult or deterioration was. And that's not always very predictable. But going back to her comment about imaging, imaging allows us to understand that process better. Yeah.
I think it's time for one more question. Oh, is there two more questions? Good.
Up on the slide behind you all, you have future cures for Alzheimer's, epilepsy and so on. The largest lettering is donated to Alzheimer's, but tonight we've heard scarce anything about it. I would be personally very appreciative if anyone or any bodies on the panel could say something about where we might possibly be getting to, if anywhere at all, with Alzheimer's.
It's a very, very fair point,
Absolutely.
actually, and yes.
There's an avalanche of Alzheimer's coming towards us as our population ages, and it's a terrible, terrible affliction. And where I see us in 10 years' time, well, actually, just take us back. We have a couple of medications that we can use now that buy us about 6 months in terms of functioning. And I think where we're heading with Alzheimer's disease, you're right to point it out, is that we are going to get to the stage in 10 years' time when we will be able to identify who's at risk of developing Alzheimer's and who isn't at risk of developing Alzheimer's by developing biomarkers, which is a fancy term for who's going to get something and who's not. And by identifying who's at risk of developing Alzheimer's, we'll be able to introduce therapies to try and prevent the disease from happening. What happens in Alzheimer's is a substance called amyloid gets laid down in the brain substance. And so hopefully we'll be able to develop drugs in people at risk to be able to prevent those amyloid plaques being laid down. There's a huge, huge amount of effort being put into Alzheimer's research, and I would have hoped in 10 years' time we'd be a lot better at treating it than we are now.
Have we got anywhere towards establishing any of those markers? Do we know? Are you confident enough to say that there's anything that we should be looking for of people of a younger age? I know half the problem is that Alzheimer's starts such a lot earlier than it shows, but is there some way that we can? Are we getting anywhere?
We are getting to the stage where we can identify patients with mild cognitive impairment, or MCI. And so there's a big study that's being carried out in Christchurch that is actually looking at this with serial clinical studies, so doing the memory tests, and these are people with just a little bit of memory problem, and it's not sure if it's just the memory problem we all get as we get older, or whether people are developing Alzheimer's disease, and they have brain scans and they have blood tests and things. So we're in the process of developing those, but I'm reasonably confident that in the next 10 years for sure we'll be able to identify people early.
You know how we all think that we're getting Alzheimer's?
Mm-hmm. Yes.
Is it actually possible to self-diagnose Alzheimer's? Or is it, I imagine that it isn't possible.
In fact, it is possible. I mean, people realise that the memory is going. I mean, all of us. I mean, my memory is shocking now, and that happens to us all. But I think Alzheimer's is a little bit more than that. And people early and in the early stages of Alzheimer's disease, in the medium stages, have insight and know that this is not right. Later on in the disease, it's different.
Thank you. I'd just like to ask a question. Why don't we get the government to help fund the Neurological Foundation? I believe sincerely that the government should be funding the Neurological Foundation.
I think that's, probably you, Alan, I think.
A
A lot of looking
Well, I
at
would
each
like,
other.
since the Neurological Foundation fund me, it would be great if the government chipped in. But, you know, the Neurological Foundation is an independent foundation. It can choose what it wants to do with its research money, and it does very well with its research money. The government does fund neurological research through the Health Research Council and the Marsden Fund. So the Neurological Foundation definitely funds a lot of the brain research, but the government also funds researchers, brain research as well. It'd be nice to have a lot more from the government, I have to admit.
Yeah, one might say, though, that the Neurological Foundation functions very well as an organization, and you wouldn't want it to actually suffer from too much government input.
Rosalie? Oh. Oh, okay.
One more, Kevin, I think.
One more question.
Yeah, I mean, it's really interesting, fascinating about the stem cell development research. I guess my question concerns whether it's a two-stage process to deal with it, which would be, for example, implanting some sort of appropriate stem cells into some part of the brain, but talking about plasticity and so forth, as you were before. Would those stem cells then, as it were, automatically take up the appropriate function in that area, or do they have to, in a sense, be, quote, reprogrammed to do so? And if so, are there ways that we would look at improving the efficiency, the speed with which that is done, so that people who were suffering and were treated in this way could recover more quickly?
So the real tricky part of any cell transplantation, whether we're talking about there's been studies been going for the last 20 years, clinical trials where they've been using fetal tissue for transplantation in Parkinson's disease and Huntington's disease, and they show that those cells do integrate, or they do connect correctly into the brain. But that's the real crucial bit. So what studies have also shown, and obviously none of these have been done in patients, with stem cells. So the only patient work we have is from the fetal tissue work. But what we know from stem cell work using animal models of disease is that they do go in there, they make the appropriate cells, and they do connect. And they do connect correctly for functional improvement. And we actually did a study about 5 years ago now where we showed with a rat model of Huntington's disease, we had animals that we didn't treat with stem cells and animals that we did treat with stem cells, and the animals that weren't treated with stem cells, progressively their motor function got worse, very similar to what you see with a Huntington's patient, and those that received stem cells, and this is a certain period after they'd started to get the symptoms of Huntington's disease, their motor function was turned back to normal. Okay, and the only way that can happen is by correct connections. And so we could then also do what we call tracing studies, where we inject a dye in one area of the brain where we hope that the cells have connected to, and we can see the body of the cell. So it goes up, gets taken up, and it gets transported up the cell body, and we could tell that was what happened. But that's a really crucial thing, and you're right, the cells have to do that properly. And the reason they can do that in an area of injury or disease is because a lot of the same signals that happen during development come up when the brain is injured as well. so which is pretty useful actually. So they actually do get the correct signals, and they seem to do a good job of replacement. If they didn't, unfortunately, what you run the risk of is epilepsy, seizure formation. So it is a very crucial thing, and it is the key component to transplant therapy, is making sure that those connections are made correctly. And in saying that, once again, it depends on what type of injury or disease you're treating as to how easy it is to do that. So if you're asking just for a nice local connection, that's not too hard. What's plagued Parkinson's disease transplantation therapy for as long as it's been going on for 20 years is that the cells die in the substantia nigra, but what they do is that the dopamine cells in the substantia nigra have this nice long pathway up to an area of the brain called the striatum. And you can't, no one's ever been able to make that long tract back up there. So what they actually do is they just actually forget about the substantia nigra, and they actually transplant the cells into the striatum, which is where the dopamine is actually released to talk to the other brain cells to control the movement. So they sort of shortcut the circuit. If you want to think about it as an electrical circuit, we can't remake that bit, so we'll just do a little bridging here. And that works because the cells that require the signal get the signal, and they just need to make a nice local connection. So, yeah.
Well, I think, Sue, that is our time up. And I can only say thank you very, very much for coming along and for your attention. Every time I sit in on one of these sessions, I feel privileged to share the stage with such great minds and brains and such wonderful people. It also surprises me to some extent that people who are so excellent at research are also so excellent at communicating to large groups. And so that surprises me. So on your behalf, I want to thank Alan and Bronwyn and Winston for their participation tonight. Can I just say that if you would like to make a donation, and I do encourage you to do this, the Foundation's appeal envelopes, which look like this, are outside, and you can get one there and make a donation, or you can donate to brain.org.nz. I thank you all for coming. I hope you've enjoyed and found this a very useful session. Thanks a million. Good night.